Hi everybody, Miss Nikki here. Welcome to Chapter 17. We're going to talk about blood today. I put a histology slide here of blood so you can take a look at it. All of these pink little discs are red blood cells. The ones that have a nucleus, that have the purple nucleus, those are white blood cells. We're going to learn the real names in just a minute. And then if you see all these tiny little dots, they could be platelets. Help if you need to clot your blood. The thing that we need to realize is there's a lot more here than just these cells. So we're going to talk a lot about the cellular components of blood, but we also need to remember that there's tons of, in the blood, there's tons of water. You have CO2 and oxygen that's being transported. From chapter 16, we know that we have hormones. There's also proteins. There's also antibodies. So there's a lot of stuff floating around in this extracellular space. This slide on the functions of blood is kind of wordy. I understand that. I just want you to take a look and realize that the majority of this you already know. So what are we transporting in the blood? Oxygen and nutrients to body cells. What kind of nutrients could we be delivering? Yep. Don't make me draw all the way out to ATP. We should know this by now, right? So because you need glucose for cellular respiration and to generate ATP, body cells need ATP in order to do action and metabolism within the body. Also, we have metabolic waste that are going to the lungs. So if I say metabolic waste to the lungs, you should be thinking CO2. And then remember, we have some waste that are going to go to the kidneys for elimination. So we could eliminate excess sodium or potassium or calcium. And then we have the hormones from the endocrine organs that are traveling in your bloodstream to get to the specific target organs. So all of this we kind of knew already. Regulation, body temperature by absorbing and distributing heat. Go back to chapter 1 in 201 and talk about sweating and shivering, right? What's the whole point of sweating and shivering? That's to help you maintain homeostasis and maintain your normal body temperature. Also, pH system using buffers. We're going to come back and talk about this. So right now, just kind of hold on. I know I mentioned it briefly when we talked about chemistry, but remember that your blood has to be around 7.35, 7.45, and we'll talk about why down the road. Adequate fluid volume for circulatory system. I want to point out that your book might use the term circulatory system, but it is not truly one of the 11 organ systems of the body. When they say circulatory, please realize that they're merging two. They're merging cardiovascular system and lymphatic system. So we'll talk more about how those two come together. It's basically all the fluid in all the extracellular fluid in your body. So that's why they call it circulatory. But again, when we did the endocrine hormones, we talked about, like aldosterone, we talked about it reabsorbing sodium and reabsorbing water and how that would affect your blood volume and your blood pressure. Blood loss. So we're going to have plasma proteins. We're going to have platelets that initiate clot formation. We'll talk about this coming up. Also, you're going to have antibodies. Complement protein, this has to do with your immune system, white blood cells as well, all the immune cells that are going to fight against a foreign invader or pathogen. So there's a lot going on here. Make sure that as you go through these functions, you don't just memorize them, but you could explain them to somebody if they asked you about them. So now we move on to talking about the composition of blood. I want to make you remember about connective tissue, hopefully. So remember, connective tissue is one of the 4 major tissues in the body, and it consists of cells and fibers. We talked about cartilage, we talked about loose and dense connective tissue, adipose tissue, bone, and all of them, if you remember, the fibers for most of those were collagen, right? We also had elastic fibers and reticular fibers, and then there were tons of different cells. Fibroblasts, right? Mast cells, tons of cells. So what's weird about blood is that blood is usually fluid, right? It's moving around. It consists of a lot of water, and then you saw the cells, the white blood cells and red blood cells and platelets that are there. But blood can form fibers. So if activated during clotting, so we have clotting factors that can activate the formation of fibers in your blood, and that helps kind of weave together or cinch back together broken blood vessels or areas of damage like on your skin. So you can activate fiber development within your blood. So it is a connective tissue. Matrix is the non-living fluid called the plasma. This is where you're going to find most of your water. We'll talk more about this in a little bit. So plasma and then formed elements. When they say formed elements, they're really talking about cells. They call them blood cells. So cells are suspended in the plasma, the formed elements, and here are the fancy words. So erythrocytes are red blood cells, leukocytes are white blood cells, and thrombocytes are platelets. And these are the guys that are involved in clotting. These are the ones that are involved in CO2 and oxygen transport, and these guys are going to be immune cells, right? Okay, let's talk about blood composition. So if I decided I was going to withdraw some blood and centrifuge it, what would it look like when we were finished? Well, it's going to separate into layers where the heavy items are going to go to the bottom and the light items are going to stay towards the top. So you can see the plasma here would be on the top. 55% of your whole blood contains plasma, and 45% contains erythrocytes, and then less than 1%, so less than 1% contains your white blood cells. So there are tons of erythrocytes, and this should make sense because they're carrying oxygen for us, right? Then our immune cells are going to be in this buffy coat, and then the plasma is going to have the antibodies, the water, hormones, any of the other items that we talked about are going to be floating here in this plasma concentration. So 55% plasma, buffy coat less than 1%, about 45% whole blood. You can see they're not super precise here, right? So one thing is that this picture doesn't really do it justice. When they call it buffy coat, it looks fluffy. That's probably the best way to explain it. It's like a white fluffy layer. So this is why we weren't really specific about that 45%. So we said 45% of your blood consists of red blood cells, and we did the little squiggle, right? Estimated. So there's something called hematocrit. So this is the percent of blood volume that is red blood cells or erythrocytes. It varies between males and females. So about 47% for males and about 42% for females, and we do have some deviation from those percentages that are still considered normal. So usually you'll hear people say about 45%. It's probably our best guesstimation of what it would be for the entire population, males and females. Any guesses as to why males would have a higher amount of red blood cells in their blood? Muscle mass. So because males have testosterone, testosterone causes increased muscle mass. If you have increased muscle mass, you have to have increased oxygen needs, and so you have to have increased oxygen being carried in your bloodstream. And remember, red blood cells, what do? They carry oxygen. So here we just talk about some of the characteristics of blood. It's sticky, it's thick, opaque, you can't really see through it. It can be anywhere from a scarlet to a dark red color. If it has high levels of O2, it's going to be a bright red. If it has low levels of O2, it's going to be a dark red. Here's that pH I mentioned earlier. This is the pH of blood. It stays within this range. If not, we're going to try to compensate for it. And we won't talk about this till the end of the semester in great detail, but basically, because you have proteins in your blood, you have to have this pH range in order for these proteins to be active. So remember, denaturation, proteins can break down if their pH and their temperature isn't optimal. So we have to make sure that our blood stays in this range, and we have ways to keep it in that range using our kidneys and our respiratory rate, and we'll talk about that at the end of the semester. So about 8% of your body weight is blood. And for males and females, you can see the average volume in liters of blood. So usually you'll see people say about 5 liters. Obviously depends on male versus female, but for the population as a whole, you'll hear a lot of people say 5 liters. So what about this plasma? A little bit more. It's usually a straw colored, it's kind of a yellowy color. It's sticky. The majority of blood plasma is water. So go back and look at some of these percentages again. So if I say all of my blood, we're saying 55% is plasma, 45% red blood cells, and then less than 1% white blood cells. Oops, white blood cells. So of this 55%, 90% is water. Over 100 different solutes can be in your blood. So you can have nitrogenous waste from metabolism. That means something that has nitrogen in it, like when you break down amino acids, creating urea. Nutrients, you can have glucose, you can have carbohydrates, you can have amino acids. You could also have fats floating around in your bloodstream. Electrolytes, sodium, potassium, chloride, calcium. This is something called bicarbonate. This is a buffer system we'll talk about later. Sorry, I keep saying that, but we're just kind of leading you into it. Obviously, respiratory gases, oxygen and carbon dioxide, and then we just spent a whole chapter talking about hormones, so you know that those are traveling as well. Those would be in the blood plasma. The other thing that we have to talk about is this protein called albumin. So albumin makes up about 60% of all of your plasma proteins, so that's quite a bit. Most of these are produced by your liver, and the function of this albumin has to do with osmosis, our favorite topic, right? So it acts as a buffer in your blood, and it basically keeps water in blood plasma or blood vessels. So we're going to come back and talk about this thing, osmotic pressure, when we get to chapter 19. So basically, remember when we talked about you have a container and you have solutes in that container, and then how does the water move? The water moves to wherever the solutes are the highest to try to dilute it out. You'd have to go back and look at, I want to say it's chapter 2 or 3 in 201. So we're going to talk about the fact that you have to have these really large proteins, these albumins, in your blood plasma in order to keep the water in the blood vessel due to this osmotic pressure. And again, we'll talk about that in chapter 19. More information for you in the form of tables in your book. So it talks about where do we have the most water, 90% of your plasma volume. They talk more about electrolytes and the sodium, potassium, the calcium, magnesium. They talk about plasma proteins, and they say they contribute to osmotic pressure and maintaining water balance in blood and tissue. And then they list them. There's albumin, there's globulins, and then there's fibrinogen. Let me get it out. Fibrinogen. And we'll talk more about this with blood clotting. So this is going to be activated, and it's going to form fiber threads that actually allow you to start the healing process or the closing of a blood clot. Continuation of that same chart talks about some of the nitrogenous substances, some that do not come from proteins, so you can also have these as a waste byproduct of cellular metabolism. There's going to be some nutrients in there. Obviously, we need glucose floating around our bloodstream, carbohydrates, amino acids, fatty acids, vitamins, cholesterol. Remember, cholesterol, the good thing cholesterol can do is help you build plasma membranes. Respiratory gases, and then hormones. Now we get to move on to that other 45% or so, and we talk about the formed elements. So we have red blood cells, white blood cells, and platelets. So white blood cells are the only complete cells, and the reason they say that is because red blood cells have no nuclei or other organelles. Basically, the nucleus has been ejected. So the only purpose of these red blood cells is to carry oxygen. So we get rid of kind of all the excess stuff. But this should tell you right here, if we eject the nucleus, can your red blood cell live forever? And hopefully you're all yelling no, right? So because the nucleus is ejected, this red blood cell cannot copy itself. So your red blood cells only live for a short period of time, and then you have to make new ones. All right, platelets here. These are really cell fragments. We'll talk about what cell they come from in a few more slides. So most of your formed elements survive in the bloodstream for only a few days. Most blood cells originate from bone marrow. So we talked about red bone marrow, I believe, back in skeletal chapter 6. Here's a nice visual for the formed elements for you. We have erythrocytes here. These are the red blood cells. We have leukocytes. These are more commonly called white blood cells. And then we have platelets. Sometimes they're referred to as thrombocytes. That's kind of the more technical name that goes with these names. All right, so if we're looking at just the leukocytes, so we're just looking at white blood cells, we have to break them down into different categories. So here we have granulocytes and agranula. And remember, if we put an A in front of something, it means not. So these guys have granules, and these guys, no granules. Here's a nice histology slide for you showing you the difference between each of the formed elements. So here we have platelets, thrombocytes, and you can see they're like little dots of cells, right? So platelets are these little pieces of a cell. They still have a purple dyed nucleus. And then here we have these larger cells that have much more complex looking structures for the nucleus. These are all white blood cells. And then they tell you specifically the different types. So this is a monocyte, this is a neutrophil, this is a lymphocyte. These are all white blood cells. And so if you see a nucleus, that has to be a white blood cell. And if it's a whole cell, if it's a little tiny fragment, it has to be a platelet. Hopefully you can see how all of the red blood cells or the erythrocytes have no nucleus. The other thing to notice is numbers. You have much higher number of red blood cells and a lower number of white blood cells just normally. If you were sick with some sort of infection, then this might be covered with white blood cells. Let's look at erythrocytes first. It's this biconcave disc, so it kind of dips in towards the middle on both sides. It's anucleated. There's no organelles, no nucleus. 97% of the cell volume is made up of hemoglobin, and this is a protein. And this protein is important in gas transport. The majority of gas that we're going to carry on a red blood cell is O2, and we do carry some carbon dioxide, but the majority is oxygen. So these red blood cells are a major factor for blood viscosity, so think thickness. If you have a high number of red blood cells, your blood's going to be thicker. More about hemoglobin. This is an oxygen-binding pigment. It binds reversibly with oxygen. This is a really important point because here's your red blood cell and it has oxygen bound. It has to go to an organ or a muscle, and what is it going to do? It's going to drop off the O2. So it has to bind reversibly. You have to allow oxygen to bind to the red blood cell, and then it has to come off of the red blood cell as easily as it bound to the red blood cell. It's responsible for the transport of oxygen in the blood. It consists of a protein called globin, bound to a red heme group. And I'll talk more about this. There's a picture coming up. So there's an iron atom in the middle of each heme, and then one oxygen molecule can bind to that heme. So each hemoglobin molecule can transport 4 oxygens. If that seems confusing, just wait. We'll talk about it on the next slide. This figure does a good job of showing you how complex the hemoglobin molecule is that's inside every red blood cell. So hemoglobin is a protein, and we can see that there's really 4 separate proteins that are coming together to form the entire structure that we call hemoglobin. So there's alpha chains and beta chains, 4 come together. Does anybody, quaternary, let me spell it right. Does anybody remember quaternary structure when we talked about proteins back in 201? So this is kind of 4th order, 4th magnitude. It has 4 proteins that come together to form this really large structure. Interesting, hemoglobin and collagen are both quaternary structures in the human body, and they're both very important. Remember, every connective tissue had collagen in it almost, and then obviously all of our red blood cells have hemoglobin. So these are 2 really important structures, and they're also 2 of the quaternary structures that we see in the body. Okay, so sorry, side note, back to hemoglobin. So at the core of every heme, and this is a heme, at the core of every heme is an iron. At the core of every heme group, the core of every globin group is a heme group. So I'll say that again. If you start inside, deep, you have an iron atom, Then you have this red heme pigment, and then you're going to have a globin protein. Hemoglobin is going to load in the lungs, and then it's going to unload in the tissues. So this makes sense, right? You breathe oxygen into the lungs, then it's going to get transported to your blood, and then it's going to be delivered to your tissue, and you're going to drop off the oxygen so that your tissues can do cellular respiration and make ATP. They just wanted to show you that there are some color-related differences due to the number of hemes and whether they're bound or not bound. So if you see oxyhemoglobin, that means there's oxygen bound to the hemoglobin. If you see deoxyhemoglobin, that means it's just hemoglobin and there is no oxygen bound to it. So these are some of the big picture things that you can be thinking about. So where would you find oxyhemoglobin? Where would you find, in my little scenario over here, where would you find oxygen bound to hemoglobin in really high concentrations? Hopefully you're telling yourself the lungs. Where would you find lots of deoxyhemoglobin? Hemoglobin that had just dropped off its oxygen. You'd find a lot of that in your tissue. Then they mentioned CO2 loading in the tissues. So remember, if you drop off CO2 in the tissues, what are you going to pick up to go back to the lungs so you can exhale? You're going to pick up CO2. So they're saying in your tissues, when you drop off that oxygen, you're also going to pick up CO2, and they call that carbaminohemoglobin. So some of your carbon dioxide is carried on a red blood cell back to your lungs in order for you to exhale. Lots of heme words, right? So this might get a little messy for you. I would probably write out all the definitions and make sure I've got all this kind of in my head the correct way, right? So we already talked about a heme group that had an iron at the center that makes up the core of globin molecules that are carrying oxygen. All right, hematopoiesis, this is blood cell formation. This means all. So hematopoiesis is if you're making red blood cells, white blood cells, platelets. So any formed cell in the body is generated by a process known as hematopoiesis. And this takes about 15 days. So this particular example, we're showing you how we make erythrocytes. So we start off with a stem cell, and remember, a stem cell can be differentiated through a pathway and become a specific cell at the end. And the one we have here, I love this name, hemocytoblast. What is it going to do? Something to do with the blood, something to do with cells, something to do with make. So it's making blood cells. So a hemocytoblast is eventually going to make erythrocytes for you. It's going to make all of your formed cells, but we'll see that in a minute. So where do you have this red bone marrow? Axial skeleton, proximal epiphyseal of the humerus and the femur. So the heads of the humerus and the femur, and then parts of your axial skeleton in your girdles, like your pelvic girdle. Now they switch over and say, hey, let's talk about erythropoiesis. So remember, hematopoiesis, I'm just going to abbreviate, hematopoiesis is all cells, all formed cells in the blood. Now we're talking specifically about erythropoiesis. So this means making red blood cells, specifically. So why do we care? Why do we have to have erythropoiesis? A couple of reasons. One, too few red blood cells leads to hypoxia. So this hypoxia, low oxygen. If you do not have enough red blood cells, you cannot carry enough oxygen. Your tissues are basically starving. They don't have enough oxygen in order to carry out cellular respiration and make tons of ATP. If you have too many red blood cells, remember that too many red blood cells makes your blood really thick think of your blood moving as thick and sluggish. That's not a great thing, right? It has to move through a tube system. And what would happen if the blood's too thick? It can get clogged. A lot of times people will relate this to changing the oil in their car, right? You have to change your oil every 10,000 miles so that it stays nice and viscous. If you have the oil too thick, it can clog up engine parts and cause problems. Same thing with the body. So there needs to be a balance between red blood cell production and destruction, and it's going to depend on hormonal controls, the supply of iron, amino acid, and B vitamins. These are kind of, you want to call them cofactors. These have to be present in order for you to make red blood cells. We also will talk about hormonal controls a little bit briefly. We'll talk about EPO, so just wait a second, we'll get there. But the last thing that I want to mention is, why do we have to make red blood cells all the time? Remember, there's no nucleus. So if there's no nucleus, they can't copy themselves, so we have to make new constantly. Here's the hormone I just mentioned, EPO or erythropoietin. This is a direct stimulus for erythropoiesis. So it's released by the kidneys in response to low oxygen. So if you have low oxygen, that's going to tell your kidneys to release EPO. And what is EPO going to do? EPO is going to stimulate hemocytoblasts to make more red blood cells. So what are some other issues that could happen? So they kind of just list here, what if you have a hemorrhage? What if you're bleeding? If you're bleeding, you have low red blood cells. So we're probably going to release erythropoietin to try to make more, right? What happens if you have insufficient hemoglobin? Remember, we had to have iron at the core of every heme. What if your diet has no iron? What if you can't process iron? Well, obviously, if you don't have the pieces to make the hemoglobin, you're not going to be able to make hemoglobin. So you have to have enough iron in order to make hemoglobin. If you don't have enough iron, you're not going to make hemoglobin. Also, something that we'll talk a little bit about is what happens if your altitude changes. What happens if the amount of oxygen available in the air that you're breathing changes? Well, then obviously that might cause you to release EPO or erythropoietin, and it might cause you to make more red blood cells. Here's our nice homeostasis teeter-totter that we see all the time in the textbook. So they're saying if you have low oxygen in your tissues due to any of these reasons, so decreased red blood cells, decreased hemoglobin, you don't have a great availability of oxygen, any of those stimuli are going to cause your kidneys to release EPO. EPO is going to go to your marrow, and it's going to tell your, specifically, they don't have it listed here, but it's going to tell your hemocytoblasts to make more red blood cells. So interesting, some athletes can actually give themselves artificial EPO. So they give themselves erythropoietin to increase hematocrit. What are we talking about here? Increase the number of red blood cells. If you're an endurance athlete and you increase the number of red blood cells, you could also increase the amount of oxygen you supply to your tissue. So if you increase the oxygen to your tissue, you might be able to increase stamina and performance. But think about this. Why is this bad? If you increase the number of red blood cells, what happens to your blood? It becomes thick. That can cause problems, right? So dangerous consequences if you increase your hematocrit too much, up to 65, and dehydration. You can have blood that becomes thick and sticky, sludge, right? And this sticky blood can pack inside blood vessels. And then that can lead to these things: clotting, stroke, heart failure. These are the dietary requirements that are necessary to make red blood cells or erythrocytes. You have to have amino acids and lipids and carbohydrates, right? You've got to be able to fuel the cell process from hemocytoblast to actual red blood cell, right? You've got to have iron because that's necessary for hemoglobin. Why? Because it's at the center of the heme group. You also have to have vitamin B12 and folic acid necessary for DNA synthesis for cell division. That goes down to this process here. Remember, the hemocytoblast is not a one-step process to a red blood cell. It goes through several stages of division and manipulation to go from a hemocytoblast to a red blood cell. The last thing I forgot to mention on the other slide was we talked about oxygen availability. I just wanted to point out if you're at a high altitude versus a low altitude. So if you're at a high altitude, you're going to have less oxygen available. And if you're at a low altitude, you're going to have more oxygen available. So this is why athletes will go and train in the environment that they're going to be performing in. They might go a month early and train. If you're living in, let's say, Louisiana, New Orleans, where you're below sea level, low altitude, and all of a sudden you're going to go perform or do some sort of competition in Colorado in the mountains, the amount of oxygen availability is going to change. And one of the things that your body could do is it could make more red blood cells. So if you had low oxygen availability and you needed to make more red blood cells, you would just increase your EPO release, and then you would increase your red blood cell concentration. I don't want you to get too caught up in the destruction of erythrocytes. I'm going to show you the image in just a second. I've kind of focused on the points that are important. So what's the lifespan of erythrocytes? They can last for 100 to 120 days, but eventually they become fragile. I mean, they're being pushed through this tube system with some force. So the hemoglobin begins to start coming apart. Macrophages are going to engulf the dying red blood cells in the spleen. So in the spleen is where you have old red blood cells, and we'll talk more about that when we get to immunity. Iron can be reused. So we might take some of this iron and recycle it back into the bloodstream so that we can use it to make more red blood cells. And then the rest of it is going to be broken down. The liver is going to secrete bilirubin, bile into the intestines. We're going to degrade the pigment, that heme group. It comes out in your feces, and the globin is going to be metabolized into amino acids. We could break those down farther, and they could be released in your urine. This gives you a nice image of what's going to happen. How would you stimulate the production of red blood cells at the top? And then it shows you here after about 100 to 120 days, the red blood cell is going to be broken down. Some of the iron can be stored. Some of it can be secreted back into the bloodstream after it goes through processing. We could break down some amino acids, put them in the bloodstream. Some food and nutrients, including the amino acids and the iron and the B vitamins and folic acid, can be reabsorbed into the bloodstream through your digestive system. So some of these things we could excrete, and then some of them we can reabsorb back into the bloodstream. I want to mention a few disorders. So we have anemia. This is if you have abnormally low oxygen carrying capacity. Usually you don't have a lot of energy, you're fatigued, you aren't flush with blood, right? So you could look pale, shortness of breath, chills. So it can be caused by any one of these items listed here. So one, blood loss. Obviously, if you're hemorrhaging, you're not going to have enough red blood cells, and then therefore you're not going to have enough oxygen carrying capacity. Two, not enough red blood cells produced. So this could be something's wrong with the processing, right? Something's wrong with your production of red blood cells. Same thing, low red blood cells, low oxygen. Too many red blood cells are being destroyed. So you could have this as well, that through that destruction process, too many of them are being tagged, or maybe they're damaged. Maybe they get damaged more quickly, and they're being processed through that pathway of breaking down and either excreting or reabsorbing, and you're just not able to make new red blood cells fast enough. These slides are just kind of more details. So blood loss, what could cause anemia, or how can blood loss cause anemia? You could have some sort of severe wound. You could also have some sort of chronic hemorrhaging anemia. So something bleeding inside of the body, like a bleeding ulcer, hemorrhoids, which are inflammation and bleeding within the hemorrhoidal veins close to the anus. Insufficient red blood cell production. So what could cause you to not make enough red blood cells? So it could be, let's say your let's say your bone marrow is being destroyed. What happens if you have some sort of tumor? What if you have a destructive tumor of your bone marrow and you have low hemocytoblasts? Remember, hemocytoblasts make red blood cells. So if you have low hemocytoblasts, you're going to have low red blood cell production. What happens if something's wrong with your EPO? For some reason, you aren't secreting enough EPO from your kidneys. The next one, pernicious anemia, is really interesting. This is a deficiency of vitamin B12. So we'll talk more about this pernicious anemia when we talk about digestion. So this is one of the vitamins that we need in order to facilitate red blood cell production. Remember, we said you need it to help DNA divide as we're going from hemocytoblast to red blood cell. And then insufficient iron. So if your iron level is low, that means your hemoglobin is going to be low. That means your red blood cell count is going to be low. And then the opposite, what happens if we have, instead of underproduction, what happens if we have overactive destruction? What happens if there's some sort of premature lysis of your red blood cells? What happens if you have faulty or abnormal hemoglobin? You remember hemoglobin is a protein. Proteins are made from DNA code, right? What if you have a genetic issue where your hemoglobins aren't exactly right? So one of those examples is thalassemias. One of your globin chains is not functioning correctly, and then you're going to make red blood cells that have deficient hemoglobin. They aren't able to carry oxygen as well. We also have sickle cell anemia. Again, a defective gene that codes for abnormal hemoglobin. They become this sickle shaped. Instead of that nice kind of shape like this, they actually form, well, that was terrible, but you got it, a sickled shape. Here's an example of a healthy or normal red blood cell, and then here is our sickled erythrocyte or red blood cell, right? And what's interesting to note is all we did was change one of the amino acids. Remember, amino acids in a chain, the sequence is important. The sequence tells you what the protein is going to look like or behave like. So all we did was change one of the amino acids, and we have a vastly different structure in our red blood cells. This red blood cell is not able to carry as much oxygen. You could also have excess red blood cells that increase blood viscosity, so polycythemia. So what happens if you have bone marrow cancer? So if you increase marrow, that'll lead to increased hemocytoblast, which will lead to increased red blood cell. This is what I talked about just a moment ago, secondary polycythemia. If there is less oxygen available, like at a high altitude, or when EPO production increases. So secondary polycythemia is not bad if it's due to climate change, right? If you went to a high altitude and all of a sudden you don't have enough oxygen, so we kick on EPO and we increase our red blood cell production, that's great when you're in the high altitude. The only time it can become bad is if you go back to if you go back to, say, a low altitude. So you've done training, you've done all your training, you won your competition thanks to your EPO making more red blood cells and carrying more oxygen. You go back to your low altitude, remember your ed blood cell is still really high. And that's great because your oxygen capacity is really high, but remember if your red blood cell count is too high, that can cause your blood to be thick. So sometimes when you see these and they list them as disorders, it's not always a bad thing. So secondary polycythemia is not a bad thing if you've gone from a low altitude to a high altitude environment and you need to perform. It can be a problem for you when you come back. You have to destroy some of those extra red blood cells, and it might take a little bit of time. And then we talked briefly about athletes can take EPO before an event to increase stamina levels, but if you take increased EPO, that's also going to cause your red blood cell count to increase. Now we get to talk about leukocytes, and you can see this is a much broader subject. We've got lots of different types. We have granulocytes and agranulocytes, and then we have 3 granulocytes and 2 agranulocytes. Don't worry so much about these numbers. I would say like these are number 1. These are the ones we have the most, and so are lymphocytes. So these are both, I guess, number 1 if we're talking about agranulocytes. All right, here's the mnemonic if you want to use it. Never let monkeys eat bananas. So that would be neutrophils, lymphocytes, monocytes, eosinophils, and bananas. I know it's not in order, so if you like it, great. If you don't, pick another one. That's fine. Okay, let's talk more about leukocytes. Remember, they make up less than 1%. Do you remember that buffy coat that we looked at? Okay, so that buffy coat makes up leukocytes, but they're critical to our defenses, and we do have a lot of them. I know when I make my students look at the microscope slide, I mean, there are just thousands of red blood cells and white blood cells, and this is a drop of blood on the slide probably. So I don't want you to think that there are only 3 or 4 like the image, microscope image depicts. There's still thousands of them. And then here's our normal range. The only reason I have this highlighted, I'm not a big numbers person, but I think it's good for examples. So let's say patient number 1 comes in and their leukocyte count is at 50,000 cubic millimeters. That's equivalent to a microliter if you're familiar with microliters. So patient comes in and has 50,000. Oh, wow, that's way off the norm. There's probably some sort of infection that's driving the production of white blood cells. Or patient number 2 comes in and let's say they have 100 leukocytes. Oh, well, that's probably bad, pretty bad too, because that means they have decreased white blood cell count. That means there's something wrong, right? Something is wrong with their hemocytoblasts, or somehow, some way, they're not producing enough white blood cells within their body. Let's look at granulocytes first. So neutrophils, eosinophils, and basophils, will go through each one. The reason they call them granulocytes is because when we stain them, we can see these little granules inside the cell. They're larger, they're shorter lived than red blood cells. They have lobed nuclei, so you might see something that kind of looks like that. They're very interesting. You can kind of see them as soon as I turn the slide, you'll have a better idea. And they can undergo phagocytosis. So if you remember phagocytosis, cellular eating, they can engulf other cells. Here are neutrophils. So neutrophils, number 1, right? Most numerous white blood cells, and they're called bacterial slayers. So they fight bacterial infections. They can basically engulf the bacteria, digest the bacteria up, and spit it out. They're attracted to sites of inflammation. Obviously, if you have a site of inflammation, it might be due to a cut, right? You might have outside bacteria able to get through your first line of defense, which is your skin. So this is a lot. 50 to 70% of all your white blood cells are used to fight bacterial infections. And you can see here what they mean by these lobed nuclei. That doesn't look like any nuclei we've seen before. And then if you notice, there's all these little granules. If they're going to be able to digest bacteria, they've got to have digestive enzymes in there. Eosinophils are pretty uncommon. They kind of have this more red staining, and they help digest parasitic worms. Hopefully these are uncommon in us, right? But in other parts of the country, you might have increased eosinophil white blood cell production. What's interesting is this last one is it plays a role in asthma. So they've shown that there are increased eosinophils in individuals that have asthma, specifically sometimes they'll call it eosinophil-induced asthma, but these eosinophils cause increased inflammation, specifically in respiratory system. So imagine if all of a sudden you have lots of inflammation in your trachea or in your bronchioles, it's going to make breathing more difficult. Basophils are rare type of leukocytes or white blood cells. They have this kind of purplish black, and you can see tons of little granules in this image, and those granules contain something called histamine. So histamine is a chemical that promotes inflammation to attract white blood cells to inflamed sites. So we're going to go through in chapter 21 kind of the steps of inflammation, but at some point you want to attract the white blood cells to the inflammation site so they can fight or digest any pathogen that might be at that site. So for right now, let's not really think about internal inflammation. Let's think about, I cut myself, I'm bleeding, and I have redness and inflammation at the site. So in that case, histamine is going to be released from your basophils, and it's going to act to attract white blood cells to the area so they can clean up any pathogens that happen to come into your connective tissue where we have a ready blood supply. Nice comparison, put them all next to each other. You can definitely see the granules everywhere, so that's why these are granulocytes, right? I would just know neutrophils are basic, and what are they trying to fight? They're trying to fight bacteria. Eosinophils, uncommon, parasitic worms, increased inflammation. Same thing with basophils, right? Release histamine. You do not have to be able to recognize them on a slide. We're going to look at them in lab, but they won't be testable. We move on to the next category, which is agranulocytes. So agranulocytes means no granules, right? So they lack granules. They're found mostly in lymphoid tissue. They are crucial to immunity, specifically these lymphocytes. We'll talk about T cells and B cells and what they do for us when we get to chapter 21 in a little bit more detail. But T cells can actively target cells that have been infected with a virus or some sort of pathogen if it's being expressed on the cell, and then B cells give rise to antibodies. So these can directly attack, and these are tagging. They're basically tagging cells that need to be destroyed. The next type of agranulocyte is a monocyte, and these differentiate into macrophages. Remember, macrophages are able to do phagocytosis, and they can protect us by engulfing viruses or bacteria or parasites and breaking them down with their digestive enzymes. These monocytes can also activate T lymphocytes and kind of help facilitate a stronger immune response. So what I would like you to think of is monocytes, macrophages, and lymphocytes. Lymphocytes, think of T cells and B cells. Here's another side-by-side image, lymphocytes, monocytes. This one's always really easy to see in the microscope. It makes kind of like a kidney shape. So monocytes are pretty easy to see. Again, there's some nice tables inside the book if you want to look at what does it look like, what's the description, how many of them do we have, how long do they last, and what's their function. So this lists all the different granulocytes. And here we have all of the agranulocytes, and then we're going to come back and talk about platelets in just a second. But for right now, we're going to talk about how do we make leukocytes. We should be seeing a trend here. Leukopoiesis is going to make leukocytes, just like erythropoiesis is the process that makes erythrocytes. So leukopoiesis is making white blood cells. They're stimulated by chemical messengers. Also, mature leukocytes can go back and say, make more. What are these chemical messengers? They've listed them here for you. Something called interleukins and colony stimulating factors. We'll talk a little bit more about interleukins when we get to chapter 21. All leukocytes originate from hemocytoblasts. We already saw this with red blood cells. So what can you say? Hemocytoblasts make red blood cells, and they make all white blood cells. I like this image because it does a good job of representing how all of the cells start off as hemocytoblasts. So we're here at stem cells, hemocytoblasts, and hemocytoblasts can be triggered to make all leukocytes, and we know also red blood cells. So it's showing you the first division of hemocytoblast, it can turn into this myeloid stem cell or lymphoid stem cell. If it turns into a lymphoid stem cell, it comes all the way down here and we make lymphocytes. If it turns into a myeloid stem cell, you're going to have pre-cells, pro-cells, and then eventually you're going to get down here and make all of your granulocytes and make one of your agranulocytes called a monocyte. So 4 of your white blood cells are made from this precursor, and 1 of your white blood cells is made from this precursor, but I want you guys to focus on this. They all come from hemocytoblasts. So how can we use this in clinical treatment? So EPO and colony stimulating factors, so erythropoietin and colony stimulating factors can be given to patients. We can stimulate bone marrow of cancer patients that are receiving chemotherapy or stem cell transplants. So if you're receiving chemotherapy, you're reducing the number of cells that divide. So dividing cells, anything that undergoes mitosis rapidly, chemotherapy can kill them, hopefully trying to kill the cancerous tumor. So if I give you colony stimulating factors, I'm trying to increase your white blood cell production. If I give you EPO, I'm trying to increase your red blood cell production. All right, some of the disorders. One, you can have leukopenia. Penia means low. So you can have an abnormal low white blood cell count. So you have decreased white blood cells, maybe due to a destructive tumor in your bone marrow. You can have leukemias. So this is where you have an overproduction of abnormal. So they're white blood cells, but they don't really work. They're abnormal. Without treatment, they're fatal. It's usually going to cause some sort of hemorrhaging, or since they don't work, you're going to have increased infection. Mononucleosis, they mention here, it's caused by a virus, the Epstein-Barr, and it increases the number of agranulocytes. So you're overproduction of these agranulocytes, and this takes energy, right? Where do you make them? You make them from hemocytoblasts, lots of cells dividing. Also, the agranulocytes will accumulate in the throat, accumulate at the site of infection. So you'll have a sore throat and fever and fatigue due to all of the inflammation and the energy that you're using to overproduce these agranulocytes. Back to this image again, just to make sure we know what we have left to talk about. So we talked about erythrocytes, we talked about leukocytes, and now we get to move on to platelets. Last group, platelets. So we kept saying these are cell fragments. Please remember these are still formed cells in blood. All right, so cell fragments of what? Megakaryocytes. So these megakaryocytes, we have a picture coming up. They're really large cells. You should get that from the mega. And little pieces are going to be pulled off, and those cell fragments are platelets. And probably the first thing to look at is this, critical to the clotting process. So basically, platelets are going to stick to, let's say, a tear. If you have a tear in a blood vessel, the platelets are going to stick and they're going to start the clotting process. So then the next bullet down says, well, why are platelets, how are they inactive? Why aren't they sticking all the time to our blood vessels? Well, we release chemicals, prostacyclin and nitric oxide, and those stop your platelets from sticking together. So basically, this is a way to kind of keep the platelets in check. If we don't need them, if there's no damage to a blood vessel, we just keep the platelets floating around in your bloodstream. As soon as there is damage, that's going to reduce these chemical messengers that were keeping them inhibited, and all of a sudden the platelets are going to be recruited and they're going to come in and they're going to start plugging the opening or the tear in the blood vessel. Last thing I want to talk about here, well, I guess I have two things. One, endothelial cells line blood vessels. Endothelial cells are the same thing as simple squamous epithelium. They just use the term endothelial cells. Instead of saying the simple squamous epithelial that line the blood vessels, they just say endothelial cells. So it's the same thing, it's just telling you location. And then the last thing here is this platelet-derived growth factor. So platelets can release this growth factor and help the blood vessel regenerate. So this is more of the regeneration process of blood clotting that we'll get to. Okay, here's our image for platelets. Here's our megakaryocyte. Here are the cell fragments. Where did megakaryocytes come from? Hemocytoblasts. So what do hemocytoblasts make? They make red blood cells, white blood cells, and platelets. So what are those? Those are all the formed cells of blood. So this stem cell is very important. Platelets age quickly. They degenerate in about 10 days. The formation of platelets is regulated by thrombopoietin. Again, we should be seeing kind of a trend here. Sometimes we'll refer to these as let's see, did I spell it right? Thrombocytes. There we go. So how do I activate thrombocyte production? I have thrombopoietin released by the liver and the kidneys.